Welcome back to lecture part 2 on metagenomics and metatranscriptomics in the Environmental Biotechnology course. We last discussed that metagenomics, as shown here, can help us identify the microbial community members and their abundance in a complex sample, such as a wastewater treatment plant, at any given point and time. However, from a microbial ecology perspective, it is more interesting to delineate the microbial functions and community interactions. To take an example of analyzing the potential functions of microbial community members identified in a wastewater treatment plant, let us look at polyphosphate accumulating organisms, or PAOs. PAOs are keystone species in wastewater treatment plants and aid in removing phosphorus from the influent wastewater, thereby preventing eutrophication. One of the most well-studied polyphosphate accumulating organisms belongs to the genus Candidatus acuminibacter. In this organism, polyphosphate serves as a storage polymer that is synthesized with the help of the PPK1 enzyme, as shown in this figure. And when required for ATP generation, the polyphosphate polymer is degraded with the help of PPK2 and PAP enzyme. In enrichment cultures, such as in the bioreactor shown, we can enrich for this organism by cycling it between feast phase, with the presence of carbon source, to famine phase with only the presence of the electron acceptor. The information generated regarding polyphosphate synthesis and degradation has also helped us identify other key organisms where these complete set of enzymes exist, namely Candidatus ozonexis and Candidatus phosphoribacter. This is a prime example of how genomic sequencing has become a cornerstone for exploring microbial communities and their potential functions. However, the short-read technologies that we have discussed so far have some limitations. Imagine a DNA sequence fragment that in its complete genome has certain repeating elements, such as shown here in the green color region. For assemblies from a short-read assembler, the computational effort is limited in its ability to assign a correct position to the green region in the assembly. However, for long-read sequencing, this is not a problem because much longer DNA fragments are passed through the sequencer and require a slightly different assembly process. Secondly, many environmental organisms have a varied GC content in their DNA, either greater than 60% or lesser than 40%. These ranges are often difficult to sequence using short-read technologies, making it difficult to answer the questions of microbial ecology framework on how these very interesting organisms survive. Long-read technologies at the workflow level, on the other hand, rely on generating much longer reads. The adapters and motor proteins for library preparation, as shown in the figure, help to sequence longer DNA fragments and thereby improve the ability to recover genomes from such organisms. As you can see, assembling complete genomes gets us one step closer to identifying potential functions of the microorganisms we sequence in a given sample. To summarize, metagenomic approaches have their strength in terms of being able to capture the complete genetic overview of microbial communities. Next, DNA in itself is a stable molecule that aids to more straightforward wet lab procedures. And this technique can be used to analyze rare or dormant microorganisms in a community. However, the shortcomings of metagenomics lie in its inability to provide information beyond the functional potential of microorganisms to capture their varied phenotypes. In this module, we present a range of multi-omics methods to investigate microbial communities. From these, in this lecture, we are focusing on the ability to describe microbial community functions using DNA and RNA. The ability to sequence the genome, either at a single gene level or metagenome level, allows for exploring the phylogeny, diversity, and hypothesize potential activity based on gene or pathway function. However, to analyze in situ gene expression, we have to rely on technologies based on RNA or proteins. In this part of the lecture, we will look at the ability to sequence RNA molecules as they are considered to be an informational molecule. As shown in the figure, the standard RNA sequencing workflow begins in the laboratory with RNA extraction followed by mRNA, that is, messenger RNA enrichment, or ribosomal RNA depletion, followed by cDNA synthesis and preparation of an adapter ligated sequencing library. The library is then sequenced on a high-throughput platform, and the final steps are computational in terms of aligning and assembling the sequenced reads to a transcriptome, quantifying the reads that overlap with the transcripts, filtering and normalizing between samples, and statistical modeling of significant changes in the expression level of individual genes or transcript between sample groups. To summarize, meta-transcriptomics approaches have strength in terms of being able to capture gene expression and patterns of the community. Next, it identifies functionally dominant microbial members. And finally, it can provide a dynamic view by reflecting the rapid changes in microbial metabolism in response to environmental stimuli. However, the limitations are that these changes in transcriptional profiles are transitionary and do not provide the complete metabolic potential of the microorganisms. Finally, the RNA is considered to be more unstable molecule when compared to DNA. Thus, to form an overall picture of microbial metabolism, it is not only important to know the genetic view of the microbial community, but also to identify the gene expression patterns and how microorganisms react to environmental stimuli. It is therefore no surprise that metagenomic and metatranscriptomic method form a coherent basis for evaluating microbial communities.